Welcome to the Demonstrating Implementation Excellence stream of SNOMED CT Expo 2024. Thank you to FAST for sponsoring this stream. My name is Sonja Ulrich and I'm the Software Development Manager at SNOMED International. I will be moderating today's session. All questions will be answered at the conclusion of the presentation. Online attendees, please use the Q&A button to type your questions to the presenter. On-site attendees, please use the microphone provided in the room. I'm now pleased to introduce Sarah Kong from FHIR, who will be presenting SNOMED CT and the veterinary extension unions and intersections for an enhanced One Health approach. Sarah, please go ahead.
Sorry, I'll start again. Hi, everyone. My name's Sarah, and I'm with the Australian eHealth Research Centre at CSIRO. And today I have the honour of actually presenting on behalf of Madonna Kemp, Donna Truran, Sam Jasinski, Andrew Bowers, Ming Zhang, and Dr. James Watson on the topic of SNOMED CT and the vet extension unions and intersections for an enhanced One Health approach. So standardized computable coded data that can be collected, retrieved, and analyzed in large volumes quickly without human interventions is essential to enable the provision of real-time surveillance on emerging public health risks due to novel viruses and/or zoonotic diseases. The team has found that variation in existing regional terminology sets does not currently support an international approach to the collection of data that can be exchanged and understood across borders. Currently, standard terminologies for human health and animal health are disjoint. There exist small handcrafted joined-up term sets, these being variable, siloed, and often lack connection between cause and effect, zoonotic and vector details, and most are constrained, distinct, special interest diseases or pathogens. So surveillance of emerging threats across human and animal populations will be enhanced by more comprehensive, inclusive standardized terminology sets covering both human and animal factors that will mean data is exchangeable and understandable across borders and information systems. Okay. So the ACDP, or the Australian Centre for Disease Preparedness, formerly known as the Australian Animal Health Laboratory, ARL, is a NATO-accredited PC4 high containment facility. This facility has specialist lab testing for animal import and export, which involves testing of animals undergoing international transport. There's emergency animal disease testing. So this is around testing for emergency terrestrial animal diseases. There's also innocuity testing, which is a range of specific and general tests to detect or exclude for adventitious agents like viruses, bacteria, fungi, or mycoplasma. And there is also a fish diseases laboratory, which provides diagnostic and research services for exotic and emerging diseases of aquatic animals, including finfish, mollusks, and crustaceans. So the team did set out to develop and implement data standards and a One Health terminology within the sample tracking and request system, STARS, and the laboratory information management system at the ACDP, with the ultimate aim being to improve data quality within collections and hopefully enabling complex analytics, bringing us a step closer to realising an integrated One Health testing and disease response system. To achieve this, they are proposing that FHIR code sets would be developed that are suited to the animal health environment, that is, a FHIR-enabled upgrade of STARS. Development of these code sets would be undertaken through the identification of content gaps and addition of new content, thereby enhancing the One Health content coverage within SNOMED CT and the vet extension of SNOMED. These code sets would be loaded and available through Onto Server. Okay, so SNOMED CT was selected because there is an existing vet extension managed by Virginia Tech, and it was felt that this promised the best options for the One Health domain in the longer term. The similarity in ontological structures, naming conventions, ECL, and the release of version schedules was also appealing, given the efforts are based on Onto Server technology and support of FHIR specifications. So the initial investigations confirmed that existing content in both SNOMED CT and the VET extension supported active and ongoing research projects within CSIRO and the ACDP, such as projects around African swine fever, lumpy skin disease virus, Japanese encephalitis, and the others listed here. And so from this respect, they were ready to go. And as part of this, other standard code systems have been considered as well. So along the journey, consideration was given to other standard code systems or taxonomies. The International Committee for Taxonomy of Viruses, or the ICTV, was also investigated. This taxonomy has much greater depth and breadth of content for viruses compared to SNOMED CT and is well researched, documented and maintained. However, it is a very different ontological or taxonomic structure. Some of the legacy data within the agents sets contained a number of cell lines. So cell line ontology and Cellosaurus were looked into. And it was found that Cellosaurus seemed to have the best fit for coverage of this type of entry. However, a cell line is not an agent, and this being contained in this particular field was more of a system issue than a terminology problem. A number of reference sites were also used to gain an understanding of the agents and or species, and it was found that the NCBI taxonomy browser is a very useful tool, particularly in terms of gaining an understanding of the Linnaean classification. And other references used were the World Health Organization for Animal Health and Atlas of Living Australia. So the next question, to map or not to map? All right, so the initial approach was to create suitable code sets for use within the enhanced systems. An initial mapping exercise was undertaken that would identify potential content rather than provide a map with exact matches or seamless forward and backward compatibility. The maps provided a design specification for selecting appropriate SNOMED and VET extension content destined for future implementation. Once candidate maps were identified, ECL was then used to construct candidate sets. In terms of processes, existing AL term sets were harvested and initial mapping efforts indicated that the scope of SNOMED CT and the VET extension would suit the STARS use case, particularly for agents, so causative agents being organisms, both human and non-human versions, and species which would be drawn from the VET extension. Breeds and animal life circumstances were also investigated by an initial mapping effort and some relevant content was identified. It should be noted that further partitioning of the organism hierarchy was necessary to separate agents and species into two distinct value sets to serve the ACDP use case. Choices were made to alter the value set membership in some cases. So for example, down the bottom there, Kingdom Protozoa is included in the agent's value set, even though it does inherit from the domain Eukarya, along with the Kingdom Animalia being assigned to the species value set. So all of this does align with the real world practice of the ACDP. Okay, so what was found along the way? So a more careful mapping of legacy data, so terms actually captured in a cumulative database at the AL was undertaken, which indicated that the scope of SNOMED CT and the VET extension content was suited for agents and species, but the maps lacked precision and were complicated by synonymy, different Linnaean or non-Linnaean approaches to naming of concepts, and there was some missingness or outdatedness in both legacy and SNOMED content. So after the initial scoping design specification mapping process, the team realised that the legacy data was important to keep, and it was difficult to determine where exactly the split should be between legacy and the design spec process. So the team undertook a more thorough process to map the legacy term lists in a more considered way to try and gain a comparative set for backward compatibility. So for species, it was found that the SNOMED or SNOVet content had 66% of equivalent or exact matches with the species set, with 32% achieving a map to an upper level parent concept. The agent set had a similar map compatibility of around 63% equivalent or exact maps, but with 15% achieving a map to an upper level concept, parent concept. With the 5% inexact maps, these were mostly concepts that the ACDP system had for agents that were considered exotic. In the end, there were 15% that weren't able to be assigned a map. Many of these were cell lines, and consideration is being given about how to record and handle these types of entries. And it is likely that these won't be recorded in the agent field. And as mentioned earlier, this is really a system architecture issue rather than a lack of content within SNOMED. With regards to the unmappables in the species set, most of these were genuine gaps and will require consideration for addition to the terminology in the future. All right, so colloquialisms, synonymy, duplicates, and missingness. These are actually factors that are contributing to inexact or no maps. So the legacy data mapping outcomes on the previous slides show that around 5% achieved an inexact map. These were mostly concepts that the ACDP system had for agents that are considered exotic or a hybrid or specific for a specific specified area, such as Nepal, Bangladesh. The difficulty with exotic forms or of agents, specifically viruses, is that what is considered exotic here may not be considered exotic elsewhere, and vice versa. Other no maps were really about data entry and user behavior rather than a genuine gap in the terminology. So maps are not necessarily a good indication that SNOMED or the VET extension lacks appropriate content or that the content needs to be reauthored. The lack of mapability tells us much about the legacy terms themselves or the intended use case and historic workflow requirements. The heritage of local legacy term lists is always variable wherever they come from and often far less formal than controlled vocabularies with well-designed logical models and editorial policies. Care needs to be taken not to include colloquialisms and local preferred terms in more formal ontology without considering the actual defining characteristics of the concept. Okay. So true missingness can only be established by thorough research and expert advice. The team noticed that some gaps in agents, particularly viruses, and some in species did exist. So most of the missingness in terms of species is at the species and subspecies levels, and this table highlights some of this. As mentioned before, the team did also look at breeds within this SNOVet content, and content there is variable with some missingness, and for animal life circumstances, they tend to have incomplete modelling, but the work within this space is continuing with some content available for utility, however none for diet. And most of this won't particularly affect this project at this current stage. So right now, the team is maintaining a list of apparently missing concepts that will be lined up for an expert panel to review and before being submitted to Virginia Tech or SNOMED International to be considered for a future release. So in both SNOMED CT and the VET extension, synonymy is variable. There is a mix of Linnaean and non-Linnaean concept names in the VET extension, and this is highlighted here and on the next slide. Some concepts that are designated as preferred terms should probably be synonyms. The mix of formal, informal names needs consideration as well, and additional synonyms should be added wherever these are understandable, reproducible, or usable. Okay. So also noted were duplicates in both SNOMED CT and the VET extension that seem to arise because there are different authoring or quality assurance approaches within and between namespace content and SNOMED International releases. So these examples here also indicate the variations of model content while still sharing synonymy. Okay. Yeah. So during the mapping processes, a number of concept matches were found to lack adequate additional synonyms. So this is something that the team would be find useful in adding to terms and to meet our use case here at the ACDP and CSIRO. All right, so now turning focus to specimens and samples and the work done here. The analysis of SNOMED content for specimen type found that it was highly pre-coordinated by comparison with the legacy term lists. This is not currently how specimen or samples are recorded in the ACDP systems. However, as the systems are in redevelopment and going through an improvement phase, there is options to explore in this space. Many concepts available within the specimen hierarchy state an anatomical site as well as a collection method, whereas the AL STARS system specifies these as separate and sometimes optional data items. Also, terms such as raw, frozen, paraffin block are required but not available as concepts in SNOMED or the VET extension. So as part of the FHIR-enabled upgrade, the team is proposing to construct an initial value set utilising the existing specimen hierarchy contained within the VET extension, as it was found that specimen type was quite well covered within both SNOMED and the VET extension. This will be implemented, and then additional concepts to meet the ACDP's needs will be collected and developed or authored iteratively as required. Content for specimen presentation was not provided for well by SNOMED CT or the VET extension. So the form of the sample being submitted to and accepted by the ACDP for processing is quite constrained and is not pre-coordinated. But this is currently under review and consideration, and there is discussion of how to handle this moving forward. The HL7 FHIR specimen resource is definitely being considered, specifically specimen type, specimen collection body site, specimen condition, specimen collection device, and specimen container additive. The table here on the right shows some of the terms from the system, and the context is not represented quite well here. So, for example, if you look down at swab could be a specimen type, or it could be a specimen collection device, or it could actually be a specimen collection procedure. So these are the types of issues that will be discussed and tackled later on. So implementation, how will it all come together? So what is being proposed is that, as the diagram shows, this is how it will be implemented into the ACDP systems. So starting at the top, SNOMED concepts will link to concept maps hosted on the ACDP Onto server. And then the concept maps will link to the code system or value sets, which are also hosted on the ACDP Onto server. The concepts in the code systems will then be used to populate terminology lists in the ACDP systems themselves. So the team is in the process of developing an interface that can be used to request new terminology entries on the ACDP Onto server. This interface is a first step in facilitating the creation of clean terminology lists based on the legacy data before full integration of the ACDP Onto server with the systems. So these images show the interface that is in its early design phase, so will likely evolve, and how users will add or request SNOMED content in the ACDP system described previously. So there will be an administration role that will review any requests that come through this interface, and we'll have those requests come through by category, so either agent, species, or sample type. And if a new request is deemed appropriate, it will have a concept created on the ACDP Onto server under the appropriate code system. And if a SNOMED concept actually exists, which is equivalent to the new ACDP concept, a concept map between the newly created concept and the SNOMED term will be created. So the link to SNOMED actually occurs via a search box where the request approver can search SNOMED for like terms based on either a concept's display value or its designations or synonyms. All right, so some future considerations for implementation. So the first being using managed service as a way to liaise and explore opportunities with Virginia Tech around veterinary extension content. The next being the content being useful beyond the Australian context and having application in international biosurveillance. And lastly, having an interest in standardising notifiable disease value sets spanning across human and animal for use in the One Health context. So further considerations for SNOMED CT and the VET extension. We would like to see a machine readable or human readable concept model, as well as an editorial guide for the VET extension, which would be most welcome. Some terms currently active in both SNOMED CT and the VET extension are outdated and could be replaced. And given our One Health perspective and the international and national enthusiasm for standard data to support the union and intersection of human, animal and environmental health, the thing that was found to be most problematic was having the human and animal content in separate releases, and a non-synchronous extension. So before I end the presentation, there are some acknowledgements that the team would like to make. Firstly, that the funding for the project was provided through the biosecurity APA initiative, and also acknowledgement to the ACDP team in informatics and specimen management, Dr. James Watson, Andrew Bowers, Sam Jasinski, and Connor Varland. And again, I'd like to acknowledge the authors of this work and active project members back home. So thank you very much.
Thank you very much, Sarah. It was a very interesting presentation. Sarah will now take questions. As a reminder, if you're in the room, please use the microphone to ask your question. Yes, John, please.
Thanks, Sarah. Great presentation. Question with the current age of PCR test and that DNA and RNA for organisms are located in the substance hierarchy. Would you find it useful to have an attribute added to SNOMED that links that DNA or RNA back to the organism hierarchy as useful in the work that's being done here?
Michael, anything? I think it's something that probably best explored just to see how it fits in with the scope of the work being done. But it's probably something I can take back to the team, and is a suggestion that if it's not hasn't been considered, can definitely go on their radar as well. So thank you for the question. Yeah.
Any other questions in the room? No? I have more of a very high level question for you. Could you elaborate, why is it important for us to understand the veterinary concepts within the context of SNOMED CT?
Yeah, so in the context, I guess, with the work that's being done with One Health, because the coverage is not just across human health, it is now spanning across human, animal and beyond. Now's the time to really start considering how it all comes together. Yeah. Us. Yeah.
And would you expect that all extension content is authored in the same way with a published concept model and thorough documentation?
Yes, I think that's what we would be moving towards and what we would hope to have, especially in the context of wanting the content to be within SNOMED. So it would be ideal, very ideal, I think, to have it following that same modelling and having that editorial guidance within. Yeah.
And do you have any concerns that this incorporating veterinary content could significantly expand the scope of SNOMED CT and its extensions?
I don't think there's too much concern. Obviously, more work needs to go into doing a further analysis, but as the team has found, there is some level of overlap. So I think starting at that point to understand how much there is in terms of duplication would give some idea of what additional content that would be, required to make it a single set of terminology to support this One Health approach. But definitely through their investigations, there has been identified that there is overlap and, cross-utility across both. So, yeah.
Yes, thank you very much. Anybody else have questions? Question? No? Okay. So then thank you very much, Sarah, again for the presentation. And with that, we will conclude this block of sessions. Online delegates, you can rejoin the next group of sessions at 15:30 Korean Standard Time through the event lobby. On-site delegates, it's time for lunch, which will now take place in the exhibition hall, where you can also view the e-posters and lightning talks. And it's your last chance to visit the exhibitors and play our passport game. Okay, see you there. Thank you very much. Bye-bye.